This video covers Advanced Higher Biology, Unit 2, Organisms and Evolution. Specifically, Key Area 1, Field Techniques for Biologists. Sub-Key Area A, Health and Safety, where we'll discuss the specific hazards and control measures used in field studies. And Subsection B, Sampling of Wild Organisms, when we'll look at ethical sampling and the different methods of detection and capture. So we discussed in our first video of Unit 1 that when we carry out practical work, whether it's lab or field-based, we need to carry out risk assessment. Well, field work has the same hazards as lab work, so the use of chemicals, heavy or hazardous machinery and equipment, contact with hazardous organisms, and lone working. Field work often has a wider range of considerations and hazards. Hazards in field work can include adverse weather conditions, difficult terrain, and the problems associated with isolation. It's more likely in field work than in lab work that the researcher could be many miles from the nearest colleague, and if they become injured, this could be dangerous. To repeat from the previous video, hazards can be objects, organisms, or chemicals which have the potential to cause harm. Risks are the likelihood of that harm arising from exposure to that specific hazard. A risk assessment involves identifying reasonable control measures to minimize the risk. In the field, these control measures may include appropriate equipment, clothing, footwear, and a means of communication. It's also really important that researchers have been appropriately trained for the conditions and the techniques they're going to be carrying out. Good sampling technique should minimize the impact on the wild species and habitats that we're going to be working in. In Unit 3, we'll discuss the 3 R's of ethical animal studies, but I thought it was important to also talk about them here. So in replacement, we replace animals with other test subjects and organisms where it is possible. We reduce the number of animals used and refine the techniques used to reduce the impact on the animals if we have to use them. Consideration should definitely be given to rare and vulnerable species and habitats that are protected by legislation. So it's very important before you start your study, while you're designing it in the pilot stage, to look at which organisms you're going to use, which ones will be appropriate, and which habitats you might find it difficult to access. When we sample a given species, we should consider which technique would be most appropriate for us to use. So point counts involve the observer recording all individuals they can see from the fixed position count location. So you can see this gentleman here, can see a yellow bird and a red bird. This can be then compared to other point count locations or with data from the same location gathered at other times. For example, the number and species of birds this gentleman will see at a given time point will differ if you sampled it an hour apart over the course of a day. Quadrats of suitable size and shape or transect can be used to sample for plants and other slow-moving or sessile organisms. So if it's not going to move, you might as well lay out a transect line and sample at regular intervals. Capture techniques such as nets and traps can be used for motile species. So these species might be difficult to observe though, so sometimes we have to use direct sampling methods like camera traps or indirect methods such as scat sampling. So we have discussed hazards in a fieldwork setting. So this can include adverse weather conditions, difficult terrain, problems associated with isolation, and contact with harmful organisms. Sampling should be carried out in a manner that minimizes the impact on wild species and their habitats. Consideration should be given to rare and vulnerable species and habitats that are protected by legislation. The chosen technique should be considered. So we're going to use point counts, where the observer records all individuals seen from a fixed point count location. It can then be compared to point count locations or from data from other times of the day. We could use quadrats of a suitable size or shape or transect lines for plants and other sessile and non-moving or slow-moving organisms. Capture techniques such as traps and nets can be used for moving mobile species. And elusive species, so those that are difficult to sample or which it might be unethical to trap and sample, can be sampled directly using camera traps or indirectly using a method like scat sampling.